And as I indicated, if you go to the website shown on the slide here, you can get directly into the WebEx, and that will allow you to view the slides on your laptop with a slight delay, I think. And that's a possibility if you'd like to be able to see some of the really interesting videos and visuals that we're going to be having on the coming slides. So to start our second session here, we're going to be moving into some of the specific techniques that are being used in single molecule and single cell detection and analysis. Our first speaker is Vasilis Vasiliou from Yale School of Public Health, and he's going to be talking about mass spectrometry-based techniques for metabolomics and imaging for environmental health research. So we're moving now towards more direct applications in the environmental health field, and we appreciate the presentation.
Well, thank you very much for the invitation and be part of this group, which is so far amazing. The talks are fascinating. One of the things that before I start, and I want to mention is we cannot go anywhere without the engineers. Single cell technologies is based on engineering, so we need to partner with them in order to advance the field. So I'm just going to start. I'm talking today not only as an investigator, but also as a chairman of a Department of Environmental Health Sciences. And essentially so we have a great interest on the exposome, and what we're trying to do is we're trying to connect the exposures, exposure assessment, exposome, in other words, effects on humans, and combine that with the epidemiology in order to make better assessments of the effects of the exposure in that. So as long as I arrived at Yale, I organized my first series of first symposium on the lifetime exposures and the exposome. We had a great speaker list in here, and we published this report into Human Genomics. And if you would like to get more information, you can go to this site, and you can all the talks have been recorded in there. So we still we're very interested on environmental influences and health outcomes. So of course we have a great interest of the metabolome. The metabolome is the closest to the phenotype and encompasses all the influence from the genome to the environment. If you want to take a look at here, this is our metabolome here. We have the gut microbiota. We heard a lot about genome, and we heard also from the first talk about gut microbiome to a certain extent. You also have the environment in terms of stress, diet, and lifestyle, but of course you have the xenobiotics that are present in all exposures, from the air pollution to water to everything. So I think you get the best, the metabolome, which is the data set of small molecules that consist in every tissue and every cell. This is what the metabolome is. So I'm going to briefly tell you a little bit of the classic metabolomics. This is very basic stuff. Untargeted metabolomics is essentially a discovery approach that aims to measure the broadest range of both endogenous and exogenous molecules, low molecular weight, less than 1500, that are present and extracted in sample without a priori knowledge of the metabolome. It is, everybody says it's a fishing expedition, it is a discovery. That's what we're going. And then this analysis can reveal novel metabolites associated with phenotype and inform pathways. So essentially what you do is you have a population, either knockout mice and wild-type mice, or you have people exposed and non-exposed, and you look at various spectrum of molecules, and then you compare those molecules, and you find what are the molecules of different, and you look at the importance of this. You can analyze that. So this is, again, the global metabolomics is simultaneous analysis of about 3,000 to 5,000 molecules that cover the range of chemical classes and metabolic pathways. The untargeted metabolomics, and especially when you're using high-sensitive hardware, allows you for high sensitivity and resolving power. In our department, we have two UPLC cutoffs from Waters, and we just got another one from Thermo, which is a high-resolution machine that we can go even lower sensitivity, higher sensitivity. So then, in addition to untargeted, you have the targeted metabolomics, where you can have a few molecules that you have identified, or 100 molecules you have identified from your untargeted analysis, and then you can go and you can quantify those molecules onto your population and either cells and so on. So analysis is optimized to increase sensitivity and resolution for each metabolite. The good thing about the targeted metabolites is you can run internal standards, and then you can also quantify all the changes. So the approach can also be used to validate the untargeted metabolomics, as I said. So the targeted metabolomics, I mean, we have right now, we have two instruments, one from Agilent and one from Waters, that we're doing the triple quad, we're doing all the analysis of these metabolites. Finally, we're going to talk about the tissue imaging using mass spectrometry, which is, again, all these techniques are not new. They're old techniques, and they're getting into the to the higher end as we move. So we have a high definition imaging system with a multi-synapse G2, and again, this was my second symposium that I organized on tissue imaging mass spectrometry, that I had people from universities and also from the industry that we talk about the tissue imaging. And this is something really amazing, that this is how this technology can enhance the single cell studies. So why the tissue imaging? It provides predictive, prognostic, and diagnostic and surrogate markers, informs the underlying molecular mechanisms of disease, reveal biomarkers, especially for drug responses and phenotypes, define a metabolotype or metabotype for each specific genotype. You can, as you see from the single cells, once you can monitor the genotype, then you can see the effects on the metabolome. It provides a means of monitor response in recurring disease, in cancers. And finally, it describes the molecular landscape in human performance application and extreme environments. So this is a technique that we have invested quite a bit, and we're working on it. And I will show you a few of these ideas in the workflow. First, let me present you the IMS workflow, which essentially you take in the tissue, and here is a brain. You take a tissue, you cut it in the cryotome, if you want to get for the small molecules, and then you take this slide and you immediately coat it with a matrix, and we will talk later about the influences of the matrix into the metabolomics. And then you send it into the mass spec. You have the laser that can really ionize molecules that can be detected, and then it can go and you can get the spectrum. And the beauty of that, this spectrum, it is converted into the imaging that, it can give you a really good idea of what is going on in the tissue. And we'll discuss what kind of applications you can have, but one thing before I go with that is, there are two things here that you have to consider. First is the resolution, and the second is the accuracy. So, or if you want to go to a single cell, you're going to lose resolution. So this is where we're trying to improve the methods right now, how you can go down to 3 microns and still have a good resolution and accuracy into those molecules. So I actually am in love with imaging mass spectrometry, and I'll tell you why, because you can do all three aspects that we are interested on. First, you can have the targeted approach. You can identify and we ran out of battery. Okay. It's very weak. Okay, that's all right. So you can see on your left-hand side, you can do the targeted approach. You can really go, let's say you want to develop, you're going to find out what are the levels of glutathione or ATP in your molecule. You can actually spot it, a little bit of the particular molecule in there, so you can train your machine and you can screen for particular molecules. Here, this example is from glutathione and ascorbic acid. And then you can also see that you can map all the areas, and then you can quantify that, because you can really have these molecules instilled into your plate, and you can look at that. So drug distribution and quantifying and localization, this is where the industry puts all the money into it, because especially we talked about drug treatments, and especially when you're using all these drugs, and I will show you one picture that we have done is how do you know that the drug that you're giving either systematically or into the tumor reaches all the site of the tumor, and this is really the fascinating thing. The other thing also is that you can do what I actually have been trained to on drug metabolism. You can identify your metabolites into the tissue, and then you can get really important information. Finally, you can also do untargeted approach discovery and biomarkers. And of course, you can do PCA analysis, you can do all the supervised statistical analysis. It's really complicated thing. It's really easy to acquire the images, but then the statistical analysis is not that easy, but we're still advancing in and getting better and better every day. So this is the three things why I think the imaging mass spectrometry is very important. Once you get it to the single cell level, it is invaluable. So again, the workflow in here, this is actually from Richard Caprioli's site, and you can actually, this is from parafilm tissue. You can section the tissue, and then you can also use trypsin, especially when you're going to look for, larger molecules, which is fragments of peptides, and essentially you can really do this mass spectrometry. This particular method is actually really being using, and I know you cannot see it very well here. This has been utilized for a lot of clinical diagnostics, especially they're trying to develop tools for diagnosis and especially for cancer. And again, I apologize, you cannot see here. This is an IMS image showing the distribution of a particular PARP inhibitor that we're doing at Yale here when we inject it at the brain. And you can see here is the injection site into the rat brain, and you can identify the molecules, how this is essentially with a drug distributed with nanoparticles, and then you can see the release of this drug on into the area of the brain, and then when you couple this along with the histology, you can see if, what exactly and what molecules are you targeting. So, and again, I really apologize that you cannot see this story, but you look at this structure in here, you can see the distribution of the drug throughout the rat brain. So this is another work that we're doing on, we are following up the tissue imaging for alcoholic liver disease, and just have some representative images here that I'm sorry, how do I get back? So, and again, you cannot see, here is the well type. This is the mice that have been treated with regular diet. This is mice that have been, livers from mice that have been treated with alcohol. And you can see, of course, the chromatographs in here, and then I want you to see the x-axis. It has completely different levels here. This is most of the 3 times. This is especially looking for the distribution of the lipids. You know that alcohol treatment induces steatosis, and essentially that's what you're trying to quantify. But as I will explain you later, this is what we're trying to use with the multi-omics, and especially when we get down to the level of what we want to do. And of course, there are a lot of questions, and I told you before, I'm really interested for small molecules and not only for the lipids, because there are a lot of questions that we need to address. For example, in liver cirrhosis, then we have the steatosis that going to fibrosis and then to cirrhosis, and you have the stellate cells which are getting activated. So the stellate cells is what they produce collagen. What really triggers, we have been working years and years to identify what really triggers this activation of stellate cells. Is the TNF-alpha, is the inflammatory responses, or it's a single molecule that comes out of alcohol? We don't know. We're trying to give these answers and what really triggers those. And of course, it's not only in liver disease, it's also in what my colleague Naftali Kaminsky is looking at, lung fibrosis and how these events are getting activated. So, with that being said, is you can also have a combination of all these techniques, including metabolomics and also the tissue imaging. This is work from my first recruitment in the department is Caroline Johnson, who came from the Scripps Institute, and essentially you can see in her study here, she has combined the metabolomics and also the tissue imaging. So FISH and SEM analysis in tumors, colon tumors, revealed that there are biofilms, bacterial biofilms, that they're accumulated only in the right side of the tumor, of the colon. So the metabolomics has revealed, as you can see, that the biofilm-associated tissue had increased polyamine metabolites. Imaging confirmed the metabolites that they were localized to the colonic mucosa where the biofilm reside. So this is, again, a combination of all this analysis and how you can really get really meaningful data of this. And now she's going further on these studies. So we're coming down to single-cell metabolomics. So far, I have introduced you just the bulk metabolomics, the tissue biofluid metabolomics, which has some limitation. Has some good advantages, but has also some limitation. It's inherently limited because the results of metabolic analysis represent really a snapshot of the multiple cell types, and also the results can be influenced by the proportion of specific cell types within the tissue. And this is really, you just get a big idea of what is going on, okay? Metabolomic analysis on single-cell population allows more precise understanding of the cellular biochemical status and how it may affect the cell phenotype. So this is some of the characteristics which essentially contribute also to the challenging or advantages. This is from a fantastic review, which I will provide it for background also reading, which shows you cell volumes, absolute amounts of biomolecules, copy number, and turnovers for transcript proteins and small molecules in single cells of E. coli, of yeast and plant, Arabidopsis thaliana, and also the human. And you can see, at least in the human, we have quite a bit of volume in here, which is really important. And of course, the number of transcripts and everything, and the number of metabolites, which are tremendous. Another thing which is really important is the turnover. When you're talking about, single cell metabolism, is the turnover and how fast it is and what conditions you are, what are you examining, especially when you want to look at the influence of environmental factors, chemicals into the cells. So you have to really quench the metabolism, you have to stop the metabolism and get meaningful data, because once you start processing it, all these enzymes still working and changing your phenotype. So the techniques that they have, I'm going to go very briefly on that. This is, of course, classic, the same thing as the mass spectrometry is the ambient ionization, which is desorption electrospray ionization, called DESI, and I'll show you some of the recent advances that they have in there. And finally, we go to MALDI, which is a matrix-assisted laser desorption ionization, which you can do single-cell resolution, where the whole surface is systematically sampled, or you can do single-cell profiling experiment, where the laser pulses and directed towards cell-specific coordinates. And of course, you can really look towards the cytosol, you can look towards the mitochondria, which is really important, also at the nucleus. Are we there yet? No, we're not. Are we getting there? We hope yes. So this is just to show you how the single cell efforts are with DESI, and I'm really briefly going to show you this. I'm not going to go to details. I'm not an engineer. Maybe our friend can talk about the engineering, but it's essentially what I want to tell you is there are a lot of labs that are working currently in developing the single-cell metabolomics using the ambient ionization. And we're still having a good results. We're not to the point yet that we can really be at the same level as the RNA-seq. We're still getting there, but this is important. So we're still in the infancy of this science, but we're moving forward. I like this slide and I wanted to show it to you because it shows you, here as you can see the single-cell mass spectrometry from a plant cell, and here you can see the same thing from the cytosolic fraction. So you can see there is a little bit more noise, but you can clearly start getting some signals. So there are a lot of challenges again in these approaches. Low abundance of molecules, low volume in most of the cases. And the other important thing is we don't have all these databases that we can utilize to identify all our MZ signals that we're getting in here. We're getting there, but it's not there yet. Now, another thing regarding the single-cell metabolomics is you don't have the luxury to go the separation first and then go. So usually all the approaches is a shotgun, and that really brings a lot of difficulties, and especially when you're coming to the point that you have the same molecules or isovariants and they give the same signal and it's difficult to distinguish. But as I said, everything is moving forward into it. This is another again from a plant cell. What essentially what you can see is the single cell spectrum in here, and then the cell back spectrum in here. This spectrum has been really purified for background noise, and it's getting better. But the point that I want to make here is that everyone, you know, we're getting much better and better in analyzing single cells in terms of metabolomics. So single cell IMS, this is again from a recent publication that indicates, and I don't know if you can see again, this is the using the laser and the matrix on the single cell, and you can start see this is the cell, the single cell in here, then you can see the single, you know, the identification of the molecules. And again, depends on how you're setting your standards and how you go in terms of the resolution. We're working with Waters in improving the capabilities of going lower in resolution. So this is actually this paper came out in January 2nd, and this is in bioarchives, and these scientists have developed the spatial single cell profiling of intracellular metabolomes. They have it identifies more than 100 metabolites in about 10,000 individual cells together. And again, this is a combination with the fluorescent microscopy. We talk about microscopy, and especially when you get down to the single cell, you really need to have something that will tell you about what is the cell and how you can distinguish. Of course, you can get the single cell population. We'll discuss that. You can get a metabolomic signature that you can really distinguish in cell population, then you can match with the RNA-seq data. But essentially with this approach, I really liked it, and I strongly encourage you to pay attention to that. And essentially what you do is you have the microscope also, you have the plate itself, you have the microscope, and you also have the IMS acquisition, and then you combine and you get these molecules. Again, we're not detecting thousands of metabolites yet. Remember, it's low abundance, low volume. Things are getting better and better as we move, and we're going forward. And as I said, another good thing about it, it's similar to the RNA-seq, and this is, again, we're in the infancy in here. So these investigators, they have coplated NIH 3T3 cells and the HeLa cells in the same tissue culture, and based on the metabolome, they can distinguish the different kind of cells in the cell culture. We hope that we can expand this into the to the tissue, and then we can get a better, meaningful things. So this is another paper that I found it very interesting, and I would really like to share it with you, that you could see just, and again, this is on HepG2 cells, this is on a single cells, but essentially in this particular case, you can apply pesticides to these cells, and then you can monitor the changes that they occur into the redox state and also in certain molecules within the cell. This is, again, a great paper that it shows the premise of this, and essentially you can show in here, this is, you have your cell in here, you have a microsyringe that you go to the single cell, of course with a microscope underneath, and then you can extract the cytosolic fraction from there, and then you can fit it to your to the mass spectrometer, and then you can get the spectrum of the metabolites and you can analyze. And these authors have shown convincingly that they can monitor changes on glutathione and reduced and oxidized glutathione, as well as other redox state measures. In addition to that, you can see also that they have create signatures of these HepG2 cells, and you can see they can really distinguish the small molecules here and the lipids within a single cell, which is really fascinating. Again, this isn't tissue culture yet. We haven't moved to the tissue, to the real tissue yet. And of course, you know, this is going to be a challenge, but it is getting there. This is now, I am trying to wrap up and say why this is important for environmental health sciences and how we can address a lot of questions. One of the questions that I have with most of my epidemiologists in our department is birth outcomes and how you can really correlate exposures with birth outcomes. This paper came out the other day in Nature, which is one of the fascinating papers, which shows genomic instability into the mother. It can result in the either preterm birth or the death of the embryos, which is included there. And what happens is there is this genomic instability, which essentially can influence mostly the female. And this is in mice, and it can cause death through the placenta to the female, but not in the male, because the male has the testosterone. So how the DNA damage, how the DNA repair, how the DNA, all the machinery of the genomic instability could trigger the death, and what is the connection? What is the connection in terms of signaling? What are the molecules which include all the signaling? This is the questions that we're trying to get together in terms of scientific questions to be addressed for environmental health sciences. Another interesting thing is, again, we know that metal exposure during the pregnancy leads to a lot of unfortunate outcomes, and also the exposure even of the babies. So how we can monitor? We don't know the mechanism by which the metals can express their toxicity into either to any of those birth outcomes in terms of mechanistic studies. So here, this paper, this is using essentially mass cytometry, which essentially you can monitor nanoparticles and you can find the silver molecules into these particular cells. So what I'm trying to tell you here is, based on this, if you can use a combination of this technology, that you can monitor metals such as lead or other compounds, and at the same time on these single molecules you can get either the metabolome or the proteomics, you can get a better sense of the mechanism and how it is going to be affected. So, challenges associated with single-cell metabolomics. As I said, low abundance of molecules to be measured. Rapid turnover rates of metabolites within the cell. This can be adjusted by quenching, stopping essentially all the process and getting into this. Matrix artifacts. You have several types. Especially on mass spectrum, on the imaging, you have to use the matrix and in order to help the ionization. So in this particular case, some of the matrices, they develop some artifacts. In other words, they generate some ionizable molecules that have nothing to do with your phenotype. Finally, and the most important here, is the limited appropriate software for integration of mass spectrometry with, microscopy images and other omics technologies. I think this is the biggest challenge. This is why we need, and I have a few slides that I can go over with you in terms of artificial intelligence and what we're doing towards this approach. So in addition to that, remember that we need more metabolome databases, we need more MZ data that we can use to identify easier instead of trying to use internal standards to get that. So this is, again, understanding the exposure types through the metabolomics, and then you can see, you can start with the phenotype, you have the metabolite type, the phenotype, and the genotype, and of course you have all these other effects that you can apply from one exposure to the internal dose, to the biological active effective dose, to early biological effect, to functional change, and to disease. So you start from your carcinogens and you go down to initiation of the tumor and then to progression in there. And of course, how you can get into the account all this, the oxidative stress, as I said, the heavy metals, the prolonged stress, the ionizing radiation, polycyclic aromatic hydrocarbons, and then endocrine receptors. So I think we need to move forward in terms of research by using a combination of sciences. And essentially, this is where the artificial intelligence and machine learning is going to be really helpful in terms of personalized interventions and looking at the effects of our environment into the biochemical, metabolic, genetic imaging, nutrition, medical, social, and other types of data. We have the population data, and then we're going to distinguish of what is going on. So this is easy to say, this is easy to create a cartoon, but how do you do that? This is really painful, and we spent quite a bit of time. We did one first, we just got accepted 2 days ago in chemical-biological interactions, which essentially is integrated multi-omics approaches to study the role of aldehyde dehydrogenase, which is an indicator of stem cells and cancer stem cells, especially for brain and colon cancer. So this is a process that we used actually cells, and we did both metabolomics, we did proteomics and RNA-seq, and we try to combine everything into using various softwares that they exist, including Metascape. And we came, actually, the data were very encouraging, and we did show quite a few pathways which are very distinct, and this was as a result of the combination of the 3 omics that we used in this particular case. And I thought it was really important. Is it good enough? Is it very effective? It took us a long time. No, it's not. So we need to move on different kind of platforms. So in that case, I have initiated a collaboration a couple of years ago with Kirill Veleskov and the Imperial College, and we're trying to develop this basis, which is essentially software that integrates all this, especially based on imaging mass spectrometry and integrated microscopy, also in RNA-seq pathology and so on, into getting better data. So this is the basis, and you can see this is a combination. You can have the omics data, additional data, and essentially you put everything in there. I don't have the time to go over to that, but we're working on expanding this into the tissue imaging, and we're trying to bring this down to a single cell level that we can be at. But at this point, we're just doing it at the tissue level. So, in conclusion, single-cell metabolomic analysis is in its infancy. It is anticipated that this state-of-the-art technology will provide unique insights into the molecular mechanism governing cell proliferation and differentiation, especially, on cases for stem cell, particular normal stem cell, how from one cell you're ending up to different cell types. And of course also in pathological situations such as cancer cells, how you can go from cancer cells, how you can treat your breast cells, and then you can have a few of them that they will survive, and those are the cancer stem cells, and how they can get down into the to the resistance. And of course, this is antibiotic resistant as well. Environmental induced changes also in cellular function and intracellular differences in susceptibility to adverse effects. Finally, we believe that the single-cell mass spectrometry will offer a better understanding of the contribution of the cell-different population in the tissue and health disease, as we heard so far from the breast cancer individual cell population. We're not there yet, but we're getting there. Thank you so much.
I think in the interest of time, we're going to have to hold questions. We'll have a panel session after lunch with all of the speakers from session 2. So we'll have some opportunities then to ask questions.